Hi, and welcome to this webinar. My name is Theresia Hallström, and I'm a Senior Application Specialist at Nanotemper Technologies. I would like to introduce you to the speaker of today's webinar, which is Dr. Vinko Misetic. Vinko studied biology at the University of Düsseldorf, and thereafter he made a PhD in the University of Heidelberg and also at the Children's Hospital in Cincinnati in the field of head and neck cancer. Thereafter, he did his postdoc in Lausanne in Switzerland in the same field. Since 2014, Vinko has been a part of the Nanotemper team, and today he will talk about solutions for plant research.
Yes, thank you, Tessa, for this nice introduction. And hello, everyone, and welcome to our small Nanotemper plant webinar. This short session will be of interest for anyone working in the field of plants or agricultural research. We will show you how we at Nanotemper Technologies are helping scientists all around the world to do good research in that field using our systems. But let's be honest, if people think of hot research topics, it's mostly about human diseases like cancer or neurodegenerative disease and how to cure them. It's usually not plants research most of the time, and it does not get the recognition and attention in society that it actually deserves. However, if we think about the future, beside virus outbreaks and wars, we have two major challenges that are inevitable. One is the steady growth of the world population, especially in developing and third world countries. And also, most probably, the climate change that goes along with it, which leads to an increase in temperatures by several degrees. As you know, Earth's space and resources are not infinite, and now we have more and more people requiring more and more food under always more challenging conditions, like increased temperatures, droughts, more famines, insufficient water supplies, and so on. And this is the reason why research on plant production, temperature resistance, pathogen resistance, environmental stress will be increasingly important and will have an increased focus in research and society. Besides that, also plant-based drugs and vaccines will become more and more important. Therefore, we will need good scientists to do good research. But of course, a good researcher needs good tools. And this is where we as a company come into play. We provide scientists with systems that enable them to perform their experiments in a high quality, fast, and efficient manner. One major topic that we cover are binding affinities, which is very important when it comes to the analysis of signaling cascades, nutrient uptake, stress signaling, and so on. And this can be performed with our monolith system, which does not require any mobilization or binding-induced change in mass or size. And this way you can analyze any given molecule combination. You can work in any buffer, even in lysate, which means that you're able to be as close as possible to the native condition of your protein. We also deal with protein stability and folding. As you know, structure and function are always connected. Institutes and companies lose a lot of time and money because they do not perform enough stability assays. Often they do activity assays, and yes, your protein might perform well in this particular assay, but due to structural instabilities, it can perform bad in other important aspects or even aggregate after a certain time. And the Prometheus allows you to analyze stability, fold-in, and aggregation of your proteins and antibodies. It's label-free, has a broad concentration range, and also here you can work in any buffer detergent. And also another system is our Dienter system, with which you can screen in a high throughput manner for potential hits and binders in a well-played format. So as you can see in this picture, we have everything available to discover a broad variety of biological processes in plants and agriculture. Especially in plant research, you can actually do a lot of things. Just think about the fact that we can work with any given molecule in any biological system. And this is just a small part of the things that we've done, but it goes from signaling to stress signaling, pathogen interactions, nutrient uptake, and also the development of therapeutical substances. Our technology can basically be applied anywhere. And I will just give you now some short examples where you can see what scientists actually do with our system. This is a very nice one where we have a huge transmembrane transporter, which is responsible for the nitrate uptake in plants that has been analyzed. And what they did was that they inserted various point mutations, and they could analyze the impact of those mutations on the nutrient transportation. You see that if you induce, for example, the point mutation in light blue, the binding gets completely abolished. And just think about when you have a really big membrane protein and a small atom binding in the pocket, that this is very hard to analyze and it's very hard to obtain any binding affinities. Our methods, however, are sensible enough to also decipher things like that. Another important aspect is the general fact that we deal with challenging samples like membrane transporters, or I don't know, GPCRs, antibodies. And here stability and sample quality is very important. So you should always make sure to work with the best sample possible, and long-term storage, assay conditions, different buffers, mutations, purification steps, they all have a crucial impact on your protein stability. In this example, we could show with the Prometheus that only even the choice of a detergent that you use for membrane proteins has a huge effect on stability. In this case, there is up to even more than 30 degrees difference depending on which detergent you use. And for things like that, it is very nice that we have a very high throughput. You only need very small amounts of sample. It's label-free, which means you can work in any buffer and use any sort of detergents or additives. And which is also nice, we can analyze stability, folding, and aggregation simultaneously. Another important aspect is variability. Actually, our methods, they have extreme variability. As already mentioned, you can perform experiments in any biological system. This means any buffer, any additive, and even directly in cell lysate. In this example here, people have measured heat stress-induced signaling directly in green plant lysate of Arabidopsis thaliana. So really directly in the PS2 system. And of course, you can imagine your protein will behave differently if you have it in its natural environment or in some artificial buffer like PBS. So you can really be as close as possible to the native conditions of your protein and analyze various systems from bacterial lysate, plant lysate, whatever. And this is a really strong point. So this is just a short outlook of what we can do. And in general, all of our methods share some common things. First is that you really get high quality data. The data that you obtain is well reproducible. You can analyze and detect things that are not possible with other methods, as seen in the examples. All of our methods are easy to use and you get your results quickly. It starts from the handling up to the software and goes on to sample optimization and the great support that we offer. In addition to that, we provide quite cost-effective solutions, as it is a small sample consumption, not too many expensive consumables, and the devices are maintenance-free and do not have any liquid handling system or any spare parts that need to be exchanged. So you get good data in a short amount of time and you save resources in regards to work time and money. So when it comes to research, clinical trace, publications, and so on, you know that it involves a lot of time, money, work, and yeah, the careers and the good being of many people depend on a positive outcome. And many institutes and companies understand that. They understand that binding and stability are important parameters and that you need good tools to make your project successful. You need good reproducible data in a short amount of time. And therefore, we now, as a globally operating company, as you can see, we have many users from both industry and academia that use our technology successfully. Also in the field of plant research, we have really many successful users. And the best thing is to try it out. So we always provide the possibility for discussions, oral presentations, and even test measurements. And when we do test measurements, we do not only use our positive controls, but we analyze your own samples. So this is really a good thing to just try it out and see what it can do for you. And if you have any questions regarding possible applications, experiments, when it comes to stability or binding, please reach out to me and we can figure out how to proceed further. So that's it. Thank you for your attention. And let's see if we got some questions.
Thank you very nice. Thank you very much, Winkel, for this very nice presentation. I have some questions. The first one is actually, won't you get a background signal when you measure in plant cell lysate?
That's a good question. So when you measuring green plant lysate, background fluorescence is something that can happen. Normally, because chlorophyll has a quite broad fluorescence range in the red wavelength area. But for this, we have a special LED channel where you don't get any fluorescence of the chlorophyll. So with this special channel, it is possible to work directly in green plant lysate with minimal or no background fluorescence.
Oh, very good. My second question is, so actually, if I have a very thermostable protein, will that be possible to analyze with the Prometheus?
Yes. So when it comes to stability, some people have really, as you said, thermostable proteins that are stable above 100 degrees. And for this, we have a special module, so-called high temperature option, that allows you to measure thermostable proteins above 100 degrees. It is also possible, so working with small glass capillaries, it is possible to see capillaries so that you don't get any evaporation, which takes 10 seconds. And then you can measure also thermostable proteins.
Ah, that's good. And the last question I have is, what type of consumables do I need to perform experiments with your technologies?
Okay. So as I said, I think in the presentation, in general, you don't need many consumables. Basically, the only thing that you need are small glass capillaries that are specialized for the experiments and can be obtained at nanotemperature. And we also have for protein labeling that you need for the binding affinity experiments. We have special labeling kits that are optimized and designed to perform the labeling very fast and easy. But of course, if people have their own established labeling methods like, I don't know, GFP fusion proteins, Cy5 labeled proteins, whatever, they can also use their own labeling strategies. So basically what's left is the capillaries.
Okay, cool. Okay, thank you very much for this very nice presentation and answering of the questions. Thank you to you, the audience, for listening. I hope you enjoyed it. If you need any more information, either contact Winkel directly or visit our homepage. There is a lot of information to get. Thank you very much.
Thank you.